Hey everybody! Today we're going to be talking about the methyl red and the Voges-Proskauer tests that we're going to be running in our lab. And these are actually two different types of assays or tests that can be done in the same medium, and they're normally used to help differentiate and classify different types of gram-negative enteric bacteria. Now, I did say again that they can be done in the same medium. This does not mean that they can be done in the same tube. So make sure that when you go to inoculate your tubes for the MRVP test, that you inoculate two tubes. And then after you've incubated them for 24, 48 hours to a week, however long we do it for, you can then just decide that your first tube is going to be the MR test, and the VP tube is going to be your second tube will be the VP tube, something like that. Now, kind of with that short introduction out of the way, let's go ahead and talk about the media basics. So with the MR component of our test here, the MR component or the methyl red component looks for mixed acid fermentation. And our Voges-Proskauer or the VP component looks to see if our bacterium is able to produce alcoholic end products such as acetone and 2,3-butanediol. And either way, whenever we add our reagents to them, if our end products are present, then the medium should turn kind of a reddish color. Now, as I mentioned, we have to add reagents to our tubes, and this is where it becomes really important that we designate one tube as our MR tube and one tube as our VP tube. So for the MR test, our methyl red test, you're going to be adding about 10 drops of methyl red. So pretty straightforward there. For the VP tube, you do have to add 10 to 15 drops of alpha-nathol. This might also say 1-nathol on it, but it's called alpha-nathol. And then afterwards, you want to add about 10 to 15 drops of potassium hydroxide, or 40% potassium hydroxide, which is often abbreviated just KOH. So just kind of be aware of that again. Basically, if you mix and match your reagents, you're going to mess up the whole thing. And another thing on our reagents, we have to wait until after they've incubated to add them. If you inoculate your samples and then add the reagents right away, you're going to mess up the whole thing. So again, make sure that you inoculate your two tubes and then wait your 24 to 48 hours or 1 week, depending on how long our incubations are. And then after you come back, then you can add your reagents, MR to the MR tubes, alpha-nathol and potassium hydroxide to the VP tube, and then you can go ahead and score them. So in terms of scoring them and looking at their results, these guys can be kind of tricky. Let's go ahead and just talk about the MR tube first. And again, like I said, red normally means positive, but here's a problem. Methyl red is red, and we're adding it to a clear tube, which means it's going to turn red no matter what. So when it comes to scoring the MR tubes, we mean it has to be very red. So for example, this kind of red is a negative, whereas this kind of red is a positive. It has to be this really deep crimson red that we're looking at here. So again, kind of orangey, negative. Deep red, that's going to be your positive. All right. Now, it's kind of the same thing with our VP setup. So with our VP results, again, you can have something like this right here, where there was no real change to the media. That's going to be a negative result for our VP reaction. Now, if you have something like this right here, where they turn kind of red at the top, that's going to be a positive result for your VP test. And kind of a note that I should have mentioned, when we add our reagent, we don't want to shake them up or anything like that. So don't shake them, just let them make a film at the top and then look at how that top changes. A note on that though, if you get something that looks kind of like this, kind of that like grayish film at the top, that is going to be a negative result. But what we can sometimes see as well is that if you get kind of a green band or a bluish band at the top, it just means that your VP reagents didn't soak into the medium. And at that point, you do have to shake it, because if you don't, you're not going to get any type of reaction. But that should really cover kind of how we score the different tubes for their different results. And again, we're really just looking for kind of a red, and normally a deep red color is a positive. I guess another note on our VP real fast, sometimes it doesn't necessarily turn this color red, it turns more of a dark brick color. That can also be a positive result. Now, in terms of inoculating our tubes, a standard broth-to-broth transfer is commonly done. If you have your sample on a plate or on a slant, you can also do a plate-to-broth or a slant-to-broth transfer as well. But that should cover what we need to know for the MR-VP test.